It's my pleasure to introduce Professor Mara Dierssen, who is very well known in this field. She's one of the world leaders, and she must have a big problem to select what to talk about in 15 or 20 minutes, but it's a great pleasure to have her here and her presentation on translational research in Down syndrome. I noticed that there's a very modest statement in the profile of Mara in the program, which says she has published a number of peer-reviewed papers on the subject. I haven't counted them, but it's a huge number.
So, here we go. Thank you very much for this very kind presentation, and I must say thank you to Peter for doing all this great job to putting us together to discuss and hopefully to get the final goal of this symposium, that is to really create a Down syndrome research force and research group. So I think this will really make a difference in the field, for sure. And well, as you see from my title, I am trying today to show you some pieces of evidence that show how we have tried to put our basic research into clinics and what were the grounds and the difficulties we encountered. And in fact, just from today, you have seen that there are so many possibilities that we really, at some point, get like confused. No? So what should we target? What should we do? Which is the best? When? So basically, we first have to make clear in our minds what do we want to rescue, and there are several possibilities. I mean, we can either go into the development of Down syndrome, which is one problem and has specific molecular alterations that are making these brains grow in an abnormal or impaired way. And probably to this end, we should look for the cause of this function failure that we then see in the adult. To what extent what we see in the adult is developmental or is functional, and we can recover part of it or not. So the questions we face from the research point of view, but also from the clinical point of view, is when should the treatment be administered? I mean, if we do it prenatally, then we will intervene on the genetic building of this altered brain. If we do it postnatally, which in our group we think is a very good moment to do this, then we will target something that is called the activity-dependent development of the brain connectivity and circuitry. And of course, we can try also, and we have to try also, to find some targets, some molecular targets that are also useful for the adult people that have already all the symptoms established. Not only that, but the problem also with many treatments, and I am, in my background, I am pharmacologist, is that we cannot be specific, region-specific with our treatments. And something that can be very good in the hippocampus may be not so good in the cortex, and vice versa. So, in fact, we are working on trying to find ways with nanomedicines and things like that to direct our treatments to specific regions in the brain. So, of course, how much has to be defined, but also how long. Can we give something acutely that really makes a difference, that is stable in time, or should we think that we will have to give all these treatments forever? So this is something that we are also trying to understand and to put forward. So, but let's look at the reality that we face, and we face to a reality in which we have many alterations in different brain regions. Most of those are important in the cerebral cortex and in some regions of the cortex like the hippocampus, but other regions in the brain are also affected, like the cerebellum, as you very well know. more than the macroscopic alterations, we are very interested in the microscopic alterations. And you know that in Down syndrome, there are specific cells that are altered in their numbers, but also the structure of the brain trees that are very important for brain communication, for how the information is processed in our brains, those are altered. And specifically, these trees of dendrites that have all these spines show morphological alterations consistently in the Down syndrome, but also in other intellectual disability populations. So this is a very good target. And this is interesting from our point of view when we look at the function of these brains, because if we have fewer or deficient spines, we will have an abnormal connectivity. And you know, connections is the most important in life, also for humans. No? And these will lead to alterations in information processing, but also storage, because the storage of information is believed to be dependent on this modification in the synaptic strength and synaptic processing. So these spines are also interesting because they reflect how plasticity is taking place. Okay? So they will show us how the synaptic plasticity processes is evolving in our models and so on. Finally, what we see is that these persons have an altered brain structure at the microscopic level that gives rise to all these phenotypes that we are observing. So, and of course I have to say, it's clear from many of the presentations today that studying Down syndrome in mice is very important. And we are where we are now in therapy thanks to them, thanks to these little mice. Why? Because you have to think that most of the new targets do not come from clinics. They do come, the concepts at least, do come from mouse models. Bill did a great job explaining chromosome 21 homologies in mice, so I will not go into that. Just let me make a little point here, because we have two ways of approaching Down syndrome through mouse models. One is creating trisomies, and this is very important to understand the pathogenetic mechanisms of the disease, but it does tell us very few things about possible candidates. So, and I will explain a little bit of old results about the TS65TN. And to try to find good candidates, we can create also single gene transgenics. Of course, the situation will be a little bit different, and we can discuss this later. But anyway, this gives us a good idea of what of these genes among all this bunch of genes that are overexpressed in Down syndrome could be a good target for therapy. Okay, so also the other question that everybody asks always in these presentations is, well, how does an intellectually disabled mouse look like? No? And of course, we cannot model the whole intellectual disability in mice. Fortunately, they don't speak, even though these do. So what we do is we select some specific tasks that show specific structural alterations. Okay? For example, this is a task in which the mouse has to find a platform to escape from the water. They don't like water, so they want to escape, and they have to use the extramace cues to make a cognitive map in their hippocampi. And to this end, what they do is they swim through the platform, and we can create a learning curve. So if I have to find the exit here, and this would be much bigger, so I would probably need much more distance and much more time to get there the first time than when I have done this several times. No? So this is the training phase, and here you see that the distance that they need to travel until they reach the platform is decreasing across sessions, and this doesn't work very well. And here they get to the best execution level. So they get to what we call the asymptotic level. What we see in the Down syndrome model is that this happens further late. Thank you very much. Thanks. Oh, that's great. Thank you. So that this happens later on, so they need double sessions, double number of sessions to get to the same level. And in the first sessions, in fact, they don't learn very much, okay? So I have put a couple of examples just to give you a flavor of how my lab looks like. And here you see a wild-type mice in the first session, so here, when he's already good. Okay, and here we see a T65 mouse trying to find the platform in the same session, in the first session. And what we see is that not only it spends more time, but the strategy that they use to find the platform is very, very different. So they are more erratic, etc. Okay, so together with that, what we see is that, in fact, the neurons of the T65TN replicate quite good the phenotypes that have been described in human Down syndrome patients. So the arbors, the dendritic trees, are smaller and less complex, and they have significantly less numbers of spines. So, you know, our hypothesis is that this altered microstructure of the dendritic trees may probably come, in part at least, from an altered development. And, you know, when we are faced with different experience during the early years of our life, social experience, cognitive experience, sensory experience, this helps our neurons to grow and to create a circuit, a connectivity. And the way that we have to test this in mice, we cannot send them to school, of course, but we can use this system that is called the environmental enrichment. That would be a kind of early intervention in mouse models. So this early intervention makes exactly this. It stimulates different domains, cognitive, sensory, motor, and visual domains. And we can check to what extent we are successful in these mice. And you know that early intervention has led to very good improvements, but in Down syndrome population, but these improvements are not as good as we would like to be, and they are also temporary. So here you see the same learning curve, and now I will show you what happens when we put these animals into environmental enrichment. And what happens is that here are the controls that go better. They need to travel less distance, but also the trisomics, look at them. They are almost as the controls are, okay? In the wild types, this is accompanied by a plasticity, by a structural plasticity. Increase in 40% of the spines is seen even 7 months after the enrichment has been done. The problem with the trisomics is that even though they get this better performance, this is not stable in time. So we lose the improvement when 4 months more or less have passed, and of course the structural plasticity is not there. So this is proof that the processes that lead to this better performance in the cognitive test are not leading to a stable change in the brain. Okay? So we wanted to see how we could solve this. And of course, we have seen that non-pharmacological tools do work, so it's good, we have to maintain them. But to what extent we can help with pharmacological tools? And there we can go for 2 types of strategies. We can go for the symptoms, trying to restore the symptoms, or we can go for etiological treatments, trying to restore the causes. And to this end, the best way is to try to normalize the expression of these altered genes. But of course, we cannot normalize the expression of all the genes that are contained in the Down syndrome critical regions. So we have to select from all these big box of genes which are the best candidates. Because these candidates, if we can normalize their expression, could be good molecules for therapy. So one favorite gene that we have in the lab is TEOC1A. We have others, but TEOC1A is really a very good molecular candidate. This is a kinase. Kinase in biology means like molecular switch on, switch off of different proteins that are activated or deactivated in terms of phosphorylation. So TEOC1A is overexpressed in Down syndrome, as you see in this Western blot. And interestingly, it's expressed very ubiquitously during development, but it's also expressed in more specific regions in the adult brain, both in mice and in humans. And it participates in very interesting functions like proliferation, differentiation, synaptic plasticity, or neurodegeneration. So what we did was we create a transgenic mouse model that only overexpressed DOCK1A. And to make a very, very, very long story very short, those mice have learning problems, and they also have these reduced complexity similar to what we saw in the TS65TN and in Down syndrome patients. The advantage of the mouse model is that you can go into the mechanism. How does this occur? And we did some primary cultures, and we could see that what happens is that the axons do not elongate correctly, and the dendrites either. So basically what happens is that the transgenic neurons are smaller, their arbors are smaller, but also the spines look more immature, elongated and more immature, and they have less spines, okay? And interestingly, when we look at the brains, and this is a very interesting experiment, wow, I will try to finish in 5 minutes. Here we have a normal mouse with fluorescent neurons, so this is not a staining. The neurons express a green protein, okay?
4 minutes.
Thank you. Oh, you said 5 to the previous one. No, it's okay. Vale. And here you see how the transgenic look like, okay? So, well, this is just the hypothesis, so I have convinced you that these alterations are led probably by overexpression of DOCK1A. So let's see if normalizing DOCK1A we can rescue the phenotype. And in fact, if we normalize the expression, we normalize the phenotype. If you compare here with here, you see that we have a normalization of the phenotypes. And also in culture, the harmine-treated neurons recover the normal phenotype. The phenotype is completely rescued, and harmine is a DOCK1A inhibitor, so it inhibits the kinase activity. So both if we normalize expression or activity of the gene, we get these results. So basically, we wanted to see if we could boost the effects of environmental enrichment with the EGCG treatment, inhibiting DOCK1A activity. So this is just to show you that environmental enrichment also works in the transgenic. Here we have another test in which this is the normal phenotype. Here you see the alteration in the transgenic and the recovery with enrichment. Interestingly, enrichment does normalize the overactivity of DOCK1A in the brain. Okay? So then we thought, okay, let's try to go further on. And we use this epigallocatechin gallate that is extract of green tea. And in fact, let me just pass very quickly through that, telling you that it was very effective in transgenics. You see, here we recover the discrimination index, and it reduces very clearly DOCK1A activity, okay? So we thought, okay, let's pair both. And what happens is that, just believe me because I cannot explain the whole graph, we can go back if you need. What happens is that EGCG tremendously potentiates the effects of environmental enrichment in the mice. So, and in fact, it changes. This is the transgenic, you remember, the wild type. Look what happens, Look what happens after EGCG plus enrichment, here compared with here, okay? So we restore the structure of the hippocampus, okay? And here you can see it very clearly. This is the wild type. You see the difference with the transgenic, and here you see the complete recovery of the hippocampal structure, okay? So we did a clinical trial with this green tea. This was a pilot, so it was only 30 people there and 30 individuals. The good news is that it's very safe, no problems at all, no secondary effect. It ameliorates significantly specific domains, cognitive domains, mainly hippocampal domains, I must say. And the effects of the treatment go back to baseline when we discontinue the treatment. So unfortunately, the effects are not maintained, at least in a 3-month treatment, okay? What was amazing is that the parents were detecting the effect very, very clearly. And I think I cannot go into the gaps and weaknesses, just, well, I think our strength is that we can go from basics to clinics, and I think you have seen an example of that. The problem is that many basic researchers are not interested in translation, I must say. And there are other many difficulties, like administrative, ethical difficulties that we have to solve, and it's very difficult. There are no common protocols for the clinical trials, and there is very small interest from pharmaceutical companies. So the idea, the gaps to be filled, is better tools for basic research, of course, better standardized criteria for clinical trials, cognitive testing, but also better cognition-related biomarkers, not only biochemicals, EEG, evoked potentials, imaging. We need this. So fortunately, I have heard very good presentation on that this morning. Still, we need to solve a lot of things, and we can discuss this later, but we don't know if sensitivity is the same in the Down syndrome population, if tolerance is the same. We don't know from the basic research. We are not giving the correct answers. We need to tell the clinicians what could be the structural changes besides the cognitive things or the LTP thing that cannot be measured then in humans. We need to give them some readouts that they can also check in patients. And this is something that we have to be prepared to do.
Mara,
time's out. Okay. Sorry, too long.
Well, I'm very...
Just let me
really
put the acknowledgements.
sorry to interrupt you, but
no.
Because it was
You
going
right.
so well, and I can't get this to work. Thank you for an absolutely wonderful presentation, and I'm sure there's people are dying to ask questions. I think we'll have to allow one question, but we are going to have a general discussion at the end, and I think Mara's brought up many important issues that we can discuss then. So is there any burning question?
Well, it's very successful if you can show that polyphenolic compound. There's many polyphenolic compound work in Alzheimer's disease. But one thing, can you measure that is where it's working? Because like others, GSK3 beta inhibitor or what, how this polyphenolic compound, can you give your mouse and can you measure the biochemically any of the components?
The advantage of the mouse is, of course, that you can do all these studies and you can do them both in peripherically and in the central nervous system, so you can compare. I couldn't explain everything because I did already explain too much, but yes, we have checked all these. We have compared it with other polyphenolic compounds. One has to separate also the antioxidant effect,
Absolutely.
but when you only have this antioxidant effect, it doesn't give rise to the same effect. And in fact, we have then tried to reverse the positive effects in mice by restoring their DNA activity. So basically what I can say is that all these studies have led us to the conclusion that, in fact, in mice, we can see it also with other DNA inhibitor compounds that are not polyphenolic, and it looks like it's quite specific. We have measured also DNA activity, as I showed you in mice, in peripheral blood of people. We have compared homocysteine levels also because they are also related to DNA activity. So basically the aim is to try to find good biomarkers, but of course we would love to have also other biomarkers, not only the biochemical biomarkers. But as I said before, as for the basic researchers, we really need to be careful to give the clinicians the information that they can then use. I mean, if we show very nice studies in the brain tissue also, only, but not in blood, the counterpart in blood, then how are we going to measure this in
because
in
green
humans?
tea is used in Parkinson's disease. There is a very group in Israel and showing the similar result in synucleinopathy and PD.
Mhm, yeah. And
Well, at this moment, I think we make a formal request to the organisers to lay on green tea in the interval. But thank you again for a
Thank
very
you.
nice presentation.
Thank you.
I'm sorry we let it run on late, but I hope.